Welcome, curious minds, to another deep dive. Today we're taking a shortcut to becoming truly well-informed about one of life's most fundamental processes. Yeah, how our genetic blueprint, our DNA, gets copied. We're sort of unzipping the fascinating world of DNA replication, pulling key insights from Robert J. Brooker's Genetics. Analysis and Principles, the seventh edition. Think of it like peering into the inner workings of this, well, unbelievably precise biological copying machine. It's running tirelessly inside, like every living cell.
Exactly. For genetic information to pass from parent to offspring, or even just from one cell to another as we grow or heal, it has to be copied first. And this process, DNA replication, it isn't just remarkably fast. It's also incredibly accurate and perfectly timed within a cell's life cycle. So our deep dive today is really going to uncover the elegant molecular mechanisms that make all of that possible.
Right. So what's our mission here? What are we trying to unpack? We're going to start by understanding the structural features that make DNA replication even possible, how it's built allows it to be copied. Then we'll journey through the actual process, how it happens in both the simpler world of bacteria and then in the more complex setup of our own eukaryotic cells. And along the way, we'll introduce you to the key proteins, the sort of unsung heroes that orchestrate this whole dance. We'll marvel at the mind-boggling accuracy of it all, and even touch on some of the clever experiments scientists use to crack this incredible code. Okay, let's dive in. The blueprint and its copying rules. A structural overview. Okay, so to really get how DNA replicates, we probably need a quick refresher on its basic structure. Picture DNA as that twisted ladder, right? The famous double helix.
That's a perfect visual, yeah. And this ladder, it's built from two long strands. Each strand is a chain of individual building blocks. We call them nucleotides. And think of each run on that ladder as being made of two halves, where each half is one of four specific chemical bases. Adenine, A, thymine T, guanine G, or cytosine C. Now, these two opposing strands, they're held together by forces like base stacking, but also by these crucial hydrogen bonds between the opposing bases. A pairs with T, G pairs with C.
And this is where it gets really, really clever for replication, isn't it? There's that fundamental rule, the ATGC rule, that dictates how they pair up. A always locks with T using two hydrogen bonds.
Two hydrogen bonds, correct. And G always pairs precisely with C using three hydrogen bonds.
Okay.
And this specific pairing, this complementarity, that's the absolute structural key, is the secret sauce for replication. Imagine that ladder unzipping right down the middle. Each separated strand then becomes what we call a template or a parental strand.
Right, a guide.
Exactly, a guide. Then, free-floating individual nucleotides, they just line up against these templates, strictly obeying that AT and GC rule, forming new hydrogen bonds. Then enzymes come along and stitch these new nucleotides together with strong covalent bonds, building the new daughter strands.
So the original double helix literally separates, and each side acts as a perfect guide for building a brand new complementary partner strand. It's almost like using a photographic negative to print a photo. You end up with an exact duplicate.
Precisely. And the magic here is that the base sequences in both of the new double-stranded molecules are identical to the original. So DNA is replicated in a way that each new copy retains the exact same genetic information as the original molecule. It has to for life to work.
You mentioned the ladder analogy. Does that mean the two sides, the two strands, run perfectly parallel, like the sides of a real ladder?
Good question. Actually, that's another crucial detail. They're anti-parallel. It's a bit counterintuitive. If you imagine one strand running up in a specific chemical direction, we call it 5' to 3', its complementary partner runs down in the opposite, 3' to 5' direction.
Okay, 5' to 3' and 3' to 5'.
Right. And this directionality, this anti-parallel nature, is incredibly important when we talk about the enzymes that build the new DNA. They're very particular about which way they read the template and which way they write the new strand.
You got it. That's going to come up again, I bet.
It definitely will.
The detective story.
Yeah.
How we know it's semi-conservative. Okay, so before scientists really nailed down this whole process, there were, three main competing ideas back in the late 1950s about how DNA might copy itself?
Yeah, it was a genuine scientific puzzle back then. The first idea was the conservative model. This suggested that the original parental DNA double helix would somehow stay completely together, intact, and then an entirely new, separate double helix would be made from scratch. Two separate entities.
Okay, one old, one totally new.
Right. Then there was the semi-conservative model. This one hypothesized that each new double helix would be a mix, a hybrid. one old parental strand paired with one newly made daughter strand.
Hmm.
And finally, there was the dispersive model. This idea was that segments of the old parental DNA and the newly made DNA would be sort of randomly mixed together, interspersed throughout both strands of the new double helix, like a patchwork quilt.
Wow. Okay. 3 distinct possibilities. So how did they solve this? How did they figure out which one was the reality?
Well, in 1958, Matthew Meselson and Franklin Stahl designed just a brilliant experiment. It's often called the most beautiful experiment in biology, actually.
High praise.
It really is. They used heavy and light isotopes of nitrogen, so 15N, which is heavy, and 14N, the normal lighter version.
Right.
Why nitrogen? Well, because nitrogen is a key component of the DNA bases, A, T, C, and G. So they could essentially label the DNA based on its weight.
Clever. So they can track the old versus the new DNA.
Exactly. Here's the setup. They grew E. coli bacteria for many generations in a nutrient broth containing only heavy 15N. This made sure that all the DNA in these initial bacteria was labeled as heavy.
Okay, starting population, all heavy DNA.
Right. Then they abruptly switched the bacteria to a medium containing only the light 14N. So as these cells replicated their DNA and divided, any newly made DNA strands would incorporate the light nitrogen and would therefore be light.
And then they used a centrifuge to separate the DNA based on density.
Right.
Heavy sinks, light floats.
Precisely. They use a technique called cesium chloride gradient centrifugation. You spin the DNA samples really fast in a tube with cesium chloride solution, and a density gradient forms. All 14N DNA, being light, would end up near the top. All 15N DNA, being heavy, would sink lower. And crucially, if a DNA molecule had one 14N strand and one 15N strand, it would be half heavy and settle right in the middle, an intermediate density.
Okay, so what do they see after one round of replication, one generation in the light medium?
After one round, all the DNA they isolated was half heavy, a single band right in the middle.
Wow, so that immediately ruled out the conservative model.
Absolutely. The conservative model predicted 2 distinct bands. One heavy, the original parent, and one light, the totally new copy. They didn't see that.
So it had to be either semi-conservative or descriptive at that point.
Correct. Both of those models could explain a single half-heavy band after one generation. The decisive moment, the real clincher, came after the second round of replication. So after about 2 generations in the light medium, now they observed 2 distinct bands. One band of light DNA and one band of half-heavy DNA.
Ah, okay. And that fits the semi-conservative model perfectly.
Perfectly, because the half-heavy DNA from generation 1 would unzip. The heavy strand would pair with new light nucleotides, staying half-heavy. The light strand would pair with new light nucleotides, becoming fully light.
Right, and the dispersive model couldn't explain that.
No. The dispersive model predicted that the original heavy nitrogen would get more and more diluted, sort of spread evenly among all the DNA strands. It would have predicted a single band, maybe slightly lighter than the first generation, but still just one band, not the 2 distinct bands they saw.
So Meselson and Stahl's experiment, elegant. It just nailed it. DNA replication is semi-conservative. One old strand, one new strand.
Exactly. A landmark experiment. It really showed us how the molecule itself ensures faithful copying.
Bacterial replication, the molecular machinery at work. Okay, so we know the principle now, semi-conservative. Let's zoom into a living cell like E. coli and see how this actually happens. Where does it all begin on that bacterial chromosome?
Right. So bacterial chromosomes are typically circular, just one big loop of DNA. And replication doesn't just start randomly. It kicks off at a very specific DNA sequence called the origin of replication. In E. coli, this specific starting spot has a name, auric.
Aurixy. Okay.
Yeah.
And it doesn't just go one way around the circle, does it?
No, that would be too slow. It's much more efficient. DNA synthesis actually starts within Oris and then proceeds in both directions at the same time.
Bidirectional.
Exactly, bidirectionally around the circular chromosome. This creates 2 points where the DNA is unwinding and being copied. We call these replication forks. These 2 forks move away from each other, like opening 2 zippers from the middle of a jacket. They eventually race around the circle and meet on the opposite side, and that completes the replication of the entire chromosome.
Okay, 2 forks moving out from Oryx, but how does that initial unwinding even start at Oris? It sounds like it needs a trigger.
It does. It begins with specific proteins called DNA proteins. When these proteins are bound to ATP, their energy source, they recognize and bind to specific short DNA sequences within ORIS. These are called DNA boxes.
DNA proteins bind to DNA boxes. Makes sense.
Yep. And this binding, along with some help from other proteins that bend the DNA, actually puts stress on the helix. It forces the DNA to twist and causes the two strands to separate, specifically in a nearby region that's rich in AT base pairs.
Why AT rich?
Because AT pairs only have 2 hydrogen bonds holding them together compared to the 3 bonds in GC pairs. So that AT-rich region is just inherently easier to pull apart to initiate the unwinding.
Ah, the weaker spot. Clever.
Exactly. Once that initial small bubble of separation is formed, the DNA proteins, with help from another protein called DNC, act like recruiters. They bring in the main unwinding enzyme, DNA helicase. DNA helicase is the enzyme that actively breaks the hydrogen bonds between the strands using energy from ATP hydrolysis. It essentially acts like a powered zipper, unzipping the DNA and making those 2 replication forks expand and move.
And you mentioned regulation earlier. There's a way the cell controls this initiation, right? So it doesn't happen too often.
Yes, very important control. There are specific short sequences within ORIX called GTC methylation sites. Another enzyme called DAM methyltransferase adds a methyl group, a small chemical tag, to the adenine base in these GTC sites. Now, for initiation to happen efficiently, these GTC sites need to be fully methylated on both strands of the DNA. But right after replication, the newly synthesized strand hasn't been methylated yet. It takes the DAM enzyme a little while to catch up. So for a short period, the DNA at the origin is methylated on the old strand, but not the new one. And this hemimethylated state actually prevents DNA from binding effectively, temporarily blocking a new round of initiation. It's a neat way to ensure the cell only replicates its DNA once per cell cycle.
That's really elegant. A built-in delay timer based on methylation.
It is. It's crucial for controlling replication.
A replication crew. Synthesizing new DNA strands. All right, so we've got the origin identified, the initial unwinding by helicase, and the 2 replication forks moving out. What happens next to actually build those new daughter strands? You said it's like a whole crew of proteins involved.
It absolutely is. It's a complex, highly coordinated operation. Let's look at the key players right there at the replication fork. So DNA helicase is chugging along, unzipping the helix, but as it unwinds the DNA, imagine twisting a rope. It creates overwinding or positive supercoiling in the DNA ahead of it. Too much of this tension could actually stop replication or even break the DNA.
Like getting knots in front of the zipper.
Exactly like that. So to relieve this stress, another enzyme called topoisomerase II in bacteria, it's often called DNA gyrase, travels ahead of the helicase. It works by making temporary double-strand breaks in the DNA, letting the strands untwist and then resealing the breaks. It's like a molecular stress relief valve.
Okay, so helicase unwinds, topoisomerase manages the tension. But the separated strands, what stops them just snapping back together? They're complementary, after all.
Good point. That's the job of single-strand binding proteins, or SSBs. As soon as the strands separate, these proteins quickly coat the exposed single strands. They act like little placeholders, preventing the strands from reannealing or reforming the double helix. They keep the template bases accessible for the copying process.
Okay, strands open, kept open. Now, how does the building start? You mentioned DNA polymerase has a limitation.
Right, here's the quirk. DNA polymerase, the main builder enzyme, cannot start a new DNA chain from scratch. It can only add nucleotides onto an existing 3' end of a nucleic acid strand.
It needs a foothold, basically.
Precisely. It needs something to extend, and that's where Primus comes in. Primus is a type of RNA polymerase. It synthesizes short stretches of RNA, usually about 10 to 12 nucleotides long, directly onto the template DNA. These short RNA pieces are called RNA primers, and they provide that necessary 3' starting point for DNA polymerase.
So it starts with RNA, not DNA.
Just a tiny piece of RNA to get things going. Once the primer is there, now the main DNA builder can take over.
Okay, finally, the star of the show, DNA polymerase.
Yes. In E. coli, the main replicative polymerase is DNA polymerase III. This is the enzyme responsible for synthesizing the vast majority of the new DNA. It catalyzes the formation of those strong covalent bonds, the phosphodiester bonds, linking one nucleotide to the next following the template sequence. It's a large, complex enzyme, a multi-subunit machine that actually wraps around the DNA template as it works. Pretty cool structure. But here's that other crucial limitation we mentioned earlier. DNA polymerase III can only add nucleotides in one specific direction. It synthesizes the new strand 5' to 3', always.
Right, the 5' to 3' directionality, and the template strands are anti-parallel.
Exactly. This creates that fascinating challenge at the replication fork.
How does the cell deal with building new strands when the two template strands are running in opposite directions, but the polymerase can only build in one direction?
This is where we get the elegant distinction between the leading strand and the lagging strand. It's a direct consequence of the 5 to 3 synthesis rule and the anti-parallel templates.
Okay, leading versus lagging, let's break that down.
All right. On one template strand, the one running 3' to 5' towards the fork synthesis is quite straightforward. A single RNA primer is made right near the origin. DNA polymerase III can then latch on and synthesize DNA continuously in the 5' to 3' direction, moving smoothly towards the opening replication fork, following right behind the helicase. This is the leading strand. It's made in one long, unbroken piece.
Okay, one continuous piece chasing the fork, that sounds efficient. What about the other strand?
The other template strand, the one running 5' to 3' towards the fork, presents the problem. Because DNA pol 3 must synthesize 5' to 3', it has to move away from the replication fork on this template.
So it's moving backwards relative to the fork's overall direction.
In a sense, yes. It can't synthesize continuously. Instead, it has to synthesize DNA in short, discontinuous bursts or segments. As the fork opens up a new stretch of template, Primus has to lay down a new RNA primer, and then DNA pol 3 synthesizes a short segment backwards, away from the fork, until it hits the primer of the previous segment. These short, discontinuous pieces are the famous Okazaki fragments, named after Reiji and Tsuneko Okazaki, who discovered them.
Okazaki fragments. Okay. How long are they, typically?
In bacteria like E. coli, they're usually around 1,000 to 2,000 nucleotides long, and each one, remember, starts with its own RNA primer. The strand is called the lagging strand.
So the lagging strand is made in these short bits and pieces. How do they eventually get joined up to form a complete continuous strand?
Right. It needs some cleanup and stitching. 3 more steps are required. First, those RNA primers have to be removed. They're only temporary starters. In E. coli, another DNA polymerase I, takes care of this. It has a special 5' to 3' exonuclease activity, basically. It can chew away the RNA primer from the 5' end ahead of it.
Okay, pol A removes the RNA.
Then, as it removes the RNA, DNA polymerase I simultaneously fills in the gap with DNA nucleotides, using the 3' end of the adjacent Okazaki fragment as its starting point. It's both removing RNA and adding DNA at the same time.
Replacing RNA with DNA.
Got it. Finally, once pol A has filled the gap, there's still a tiny nick, a break in the sugar phosphate backbone between the newly synthesized DNA and the previous Okazaki fragment. That final connection is made by DNA ligase. It acts like molecular glue, catalyzing the formation of that last phosphoester bond, sealing the nick and creating a continuous, unbroken lagging strand.
Wow. Leading strand, continuous lagging strand in fragments that get cleaned up and stitched together by pol-IM and ligase. It's quite a process. Do these enzymes, like helicase, primus, polymerase, just float around and find each other?
That's what people initially thought, but it's much more elegant. What's truly fascinating is they don't operate independently. They form a coordinated machine. The DNA helicase and the primus are actually physically bound together. This complex is called the primosome.
Primosome, okay.
Then, this primosome associates with 2 DNA polymerase III holoenzymes, one for the leading strand, one for the lagging strand, to form an even larger, highly efficient complex called the replisome. It's like the whole replication factory in one unit.
2 polymerases working together.
Yes, a dimeric DNA polymerase. They move together as a unit along with the helicase. Now, to make this work, especially for the lagging strand polymerase, which needs to synthesize backwards, the lagging strand template actually gets looped out. This looping allows the lagging strand polymerase to synthesize 5' to 3', but still physically move in the same overall direction as the leading strand polymerase and the helicase at the fork. It's a clever bit of molecular choreography.
Looping the lagging strand. Okay, that makes sense for keeping the machine together. Amazing coordination.
It dramatically improves the efficiency and speed of the whole process.
So where does this all stop? On that circular bacterial chromosome. How does it know when it's done?
Good question. On the opposite side of the E. coli chromosome, directly across from Oryx D, there are specific DNA sequences called termination sequences, or TER sequences. There's a specific protein called TUS, terminus utilization substance, that binds tightly to these TER sequences. When a replication fork moving around the circle encounters a TUS protein bound to a TER site, it effectively acts as a roadblock, stopping the movement of the helicase and thus the fork.
Like a one-way gate.
Pretty much. There are multiple terse sites oriented in opposite directions, creating a sort of trap region where the two converging replication forks will eventually meet and stop. Replication ends when these oppositely moving forks collide, usually within this termination region.
And then you have two separate circular chromosomes.
Almost. Sometimes after replication is complete, the two newly formed circular DNA molecules can end up physically interlinked, like two rings in a chain. These interlinked molecules are called catenanes.
Catenanes, okay, tangled rings. How do they get separated?
That's where our friend topoisomerase II, DNA gyrase, comes back into play. It makes a temporary double-strand break in one of the circles, passes the other circle through the break, and then reseals it. Problem solved. The two daughter chromosomes are now separate and ready for cell division.
That enzyme really is versatile. Unwinding stress, separating finished chromosomes.
Absolutely critical throughout the process.
This level of detail is incredible.
Hmm.
How did scientists figure out the roles of all these different proteins? DNA, helicase, primase, pol first ligase, tusses. It seems impossibly complex.
It was a huge challenge, and a major breakthrough came from genetics, specifically from studying mutants. Researchers worked primarily with E. coli and looked for bacteria that had defects in DNA replication. Now, DNA replication is essential for life, right? So a mutation that completely breaks it is usually lethal. The cell can't grow or divide.
Right, dead cells don't tell you much.
Exactly. So the really clever approach was to look for conditional mutants, specifically temperature-sensitive or TEATS mutants. These ATES mutants carried a mutation in a replication gene, but the resulting protein was only defective at a higher non-permissive temperature, say 42 degrees. At a lower permissive temperature, like 30 degrees, the mutated protein could still function more or less normally, allowing the cells to grow.
Ah, so you can grow them normally, then shift the temperature to see what breaks.
Precisely. Scientists would expose bacteria to mutagens to create random mutations, then grow colonies at the permissive temperature. They'd then use a technique called replica plating to copy these colonies onto two new plates. One incubated at the permissive temperature and one at the non-permissive temperature. The rare colonies that grew at 30 degrees C but failed to grow at 42 degrees C were the temperature-sensitive mutants they were looking for.
Okay, so they find the feces mutants. How does that tell them about specific replication jobs?
They'd then take these anti-mutants and test them specifically for DNA synthesis defects at the non-permissive temperature. Some mutants showed a very quick stop in DNA synthesis as soon as they were shifted to the high temperature. These were called rapid stop mutants. This suggested a defect in a protein directly involved in the ongoing synthesis process itself, like a subunit of DNA polymerase III or helicase.
Makes sense. If the main engine breaks, everything stops fast.
Right. Others were slow stop mutants. These cells could finish the current round of replication that was already underway when the temperature shifted, but they couldn't start a new round.
Ah, so that points to problems with initiation.
Exactly. Defects in proteins needed to start replication at the origin, like the DIA protein we talked about.
So by characterizing when replication stopped immediately or after finishing a round, they could start assigning functions to the genes identified by these ITSEs mutations.
Yes. It was painstaking work, a sort of brute force genetic screen, but it was absolutely instrumental in identifying the dozens of different proteins involved in bacterial DNA replication and figuring out what each one actually does. It was genetics revealing biochemistry.
The amazing accuracy of replication. Okay, let's talk about accuracy. You mentioned DNA polymerase III in E. coli. It's incredibly fast, like 750 nucleotides per second. That sounds like a recipe for disaster, for making tons of mistakes. But the source material, Brooker's book, says the error rate is incredibly low, only about one mistake per 100 million nucleotides incorporated. That's mind-blowing fidelity. How is that even possible?
It is truly remarkable, isn't it? And it's not just one mechanism. It's achieved through a sophisticated, multi-layered system, like a series of quality control checkpoints.
Okay, what's the first layer of defense?
The first layer is simply the basic chemistry, the inherent stability of base pairing. The hydrogen bonds between a correct GC pair, 3 bonds, or an AT pair, 2 bonds, are just physically and chemically much more stable than any incorrect mismatched pair, like A with C or G with T. This fundamental preference for correct pairing provides a baseline level of accuracy, limiting errors to maybe about 1 in every 1,000 nucleotides. Still not great, but a start.
Okay, so basic chemistry helps. What's the next level?
The second layer involves the structure of the active site of DNA polymerase itself. The part of the enzyme where the new nucleotide is actually added is shaped very precisely. It's like a highly specific lock. It preferentially binds and catalyzes the addition of a nucleotide only when it forms the correct base pair with the template strand. If an incorrect nucleotide tries to enter the active site, the mismatch causes a slight distortion in the shape of the DNA helix right there. This distortion physically prevents the incorrect nucleotide from fitting properly into the active site and triggering the necessary conformational change in the enzyme, a process called induced fit that's required for the chemical reaction to occur.
So the enzyme itself performs a fit check. It won't catalyze the reaction if the geometry is wrong.
Exactly. It's a crucial structural checkpoint. This mechanism dramatically improves accuracy, reducing the error rate down to somewhere in the range of 1 in 100,000 to 1 in 1 million errors. Much better, but still not quite the final number.
Okay, that's a huge improvement. What's the final, most impressive layer? The one that gets us to 1 in 100 million.
The third layer is proofreading, and it's arguably the most critical. DNA polymerase isn't just a builder, it's also its own editor. Most replicative DNA polymerases, including Pol III and E. coli, have an additional enzymatic activity called 3' to 5' exonuclease activity.
Exonuclease, meaning it cuts nucleic acids from the end.
Yes, specifically from the 3' end of the newly synthesized strand. If the polymerase accidentally adds an incorrect nucleotide, it can usually detect the resulting mismatch distortion. When it detects a mistake, it pauses, shifts the 3' end of the growing strand out of the polymerase active site and into this separate exonuclease active site. The exonuclease then acts like a backspace key or an eraser. It snips off the incorrect nucleotide. Then the strand shifts back to the polymerase site and synthesis resumes with the correct nucleotide being added.
Wow. So it double-checks its own work and fixes mistakes on the fly.
Exactly. This active proofreading function is incredibly effective. It catches most of the errors that slip past the first 2 checkpoints, bringing the overall error rate down to that final, amazing level of about 1 mistake per 100 million nucleotides.
Incredible. 3 layers. Base pair stability, polymerase active site geometry, and active proofreading.
That's the trifecta for high-fidelity replication.
And you mentioned speed earlier. How does it manage to be so fast and stay attached to the DNA? Does this proofreading slow it down?
That's where another feature comes in. Processivity. DNA polymerase III is a highly processive enzyme. This means it doesn't just add one nucleotide and then fall off the DNA template. It stays tightly associated with the template and slides along, adding thousands or even tens of thousands of nucleotides before dissociating.
It holds on tight.
Right. There's a specific subunit of the DNA polymerase III holoenzyme in E. coli. It's the beta subunit that forms a ring-like structure. It acts as a sliding clamp. This clamp encircles the DNA double helix and tethers the main polymerase core enzyme to the template. This allows for continuous high-speed synthesis without the enzyme constantly falling off and having to reattach, which would dramatically slow things down.
The sliding clamp. So that's key for both speed and efficiency. It seems like every aspect has been optimized.
It really does. It's an incredibly refined molecular machine.
Eukaryotic replication. Similarities, complexities, and telomeres. Okay, we spent a lot of time on bacteria like E. coli. Now let's shift focus to eukaryotic cells, like the ones in plants, animals, fungi. Are their basic principles the same, or does it get, well, a lot more complicated?
Well, many of the general features are similar. Eukaryotes definitely still use DNA helicases, topoisomerases, single-strand binding proteins, primases, DNA polymerases, and DNA ligases. The fundamental chemistry and the semiconservative mechanism hold true. However, eukaryotic replication is overall substantially more complex. And there are several reasons for this.
Like what?
First, eukaryotes have much, much more DNA than bacteria. And it's not in a single circle. It's organized into multiple, long, linear chromosomes. Second, this DNA isn't naked in the nucleus. It's tightly packaged with proteins, mainly histones, into a complex structure called chromatin, specifically into repeating units called nucleosomes. Replication has to deal with unpacking and retacking this structure.
Why the DNA is wrapped around those histone spools.
Exactly. And third, the regulation of DNA replication is much more intricately tied to the overall cell cycle in eukaryotes, ensuring DNA is copied only once during the S phase before the cell divides.
Okay, linear chromosomes, chromatin packaging, tighter cell cycle control. Let's start with the size. Given our really long linear chromosomes, how do our cells manage to copy all that DNA in a reasonable time frame? One origin wouldn't cut it, surely.
Absolutely not. If we only had one origin per chromosome, replication would take days or weeks. So the key adaptation in eukaryotes is having multiple origins of replication scattered along the length of each linear chromosome.
Any starting points?
Many starting points, hundreds or even thousands per chromosome. Replication initiates simultaneously or nearly simultaneously at many of these origins and proceeds bidirectionally from each one, creating multiple replication bubbles.
Bubbles that expand and eventually merge.
Precisely. This was shown beautifully in experiments back in 1968 by Joel Huberman and Arthur Riggs. They used radioactive labeling pulses and saw these multiple replicating segments along the DNA fibers. Eventually, all these replication forks meet, and the entire chromosome is duplicated.
Do these origins look like the ORC sequence in bacteria?
It varies. In simpler eukaryotes like yeast, the origins, called ARS elements, autonomously replicating sequences, are defined by specific DNA sequences, somewhat like ORIC. But in more complex eukaryotes, including animals, the specific DNA sequence seems less important than the overall chromatin context. Origins are often found in regions with specific chromatin structures or histone modifications, suggesting it's more about accessibility and the local environment than just a short DNA code.
Interesting. So structure matters as much as sequence for origins in us. How about initiating replication? Is it still DNA-like proteins?
It's analogous, but involves a different, more complex set of proteins. Initiation in eukaryotes involves the assembly of a large protein complex called the pre-replication complex, or pre-RC, at each origin. This happens during the G1 phase of the cell cycle, well before DNA synthesis actually starts.
G1 phase, the prep phase.
Right. A key component is the origin recognition complex, or ORC. It binds to the origin DNA and acts as a landing pad for other proteins. ORC recruits other factors like CDC6 and CDT1, and critically, these help load the MCM helicase onto the DNA. MCM stands for minichromosome maintenance. This loading of the MCM helicase is a crucial step called DNA replication licensing. It essentially marks that origin as ready to fire, but keeps it inactive for now.
So it's licensed in G1, but doesn't start replicating yet.
Correct. The activation is tightly controlled and only happens when the cell enters S phase.
And what triggers the start in S phase?
As the cell transitions into S phase, specific protein kinases enzymes that add phosphate groups become active. They phosphorylate key components of the pre-RC and other replication factors. This phosphorylation acts like a switch. It causes some initiation factors like CDC6 and CDT1 to be released or degraded, preventing relicensing within the same cycle, and it recruits the actual DNA polymerases and other necessary factors to the origin, activating the MCM helicases. The MCM helicases then start unwinding the DNA, and bidirectional replication begins from that licensed and now activated origin.
A much more elaborate control system than in bacteria. Makes sense given the complexity. What about the polymerases? Are they the same as pol-1 and pol-3?
No, eukaryotes have a whole different and much larger set of DNA polymerases.
Yeah.
Mammalian cells, for instance, have over a dozen different types, each with specialized roles.
Over a dozen, wow. Which ones do the main replication job?
4 are primarily involved in replicating the bulk of the nuclear DNA: polymerase alpha, delta, epsilon, and gamma. DNA polymerase gamma is dedicated to replicating the DNA found in mitochondria.
Okay, so gamma handles the mitochondrial DNA. What about the nucleus?
In the nucleus, it's a team effort involving alpha, delta, and epsilon. DNA polymerase alpha works in conjunction with primus. Together, they synthesize a short RNA-DNA hybrid primer, about 10 RNA nucleotides followed by maybe 20-30 DNA nucleotides to get both the leading and lagging strands started.
So pol-alpha primus lays down the initial primer. Then what?
Then a crucial event called polymerase switching occurs. Pol-alpha primus has low processivity, meaning it falls off easily, so it dissociates and other more processive polymerases take over. It's generally thought that DNA polymerase epsilon is the main workhorse for synthesizing the continuous leading strand, and DNA polymerase delta is primarily responsible for synthesizing the discontinuous lagging strand Okazaki fragments.
Epsilon for leading, delta for lagging, after pol-alpha starts things off. Got it.
Yep. And there are also specialized translesion synthesis, or TLS polymerases, like eta, kappa, iota. These are like backup polymerases. If the main replicative polymerases, epsilon or delta, encounter a damaged section of DNA template, they might stall. These TLS polymerases are less accurate, but they have the ability to synthesize across certain types of DNA damage, allowing replication to continue, albeit potentially introducing a mutation. It's like a temporary patch to get past a roadblock.
A trade-off. Bypass the damage but risk an error.
Sometimes that's necessary to complete replication.
Okay, so different polymerases. How are the RNA primers removed in eukaryotes? Is it still polymerate in E. coli?
Another key difference. Eukaryotes primarily use a different mechanism. Instead of polo, an enzyme called flap endonuclease, or FEN1, plays the major role, along with DNA polymerase delta.
Flap endonuclease. How does that work?
As DNA polymerase delta synthesizes an Okazaki fragment, it runs into the RNA primer of the preceding fragment. Instead of stopping, it actually pushes aside the initial part of the RNA primer, creating a little single-stranded RNA flap.
Okay, it displaces the primer.
Right. Then FEN1 comes in and specifically recognizes and cleaves off this RNA flap. Sometimes a longer flap is created, which needs help from another nucleus called DN2 first, but FEN1 makes the final cut. This process might repeat a few times until the entire RNA primer is removed. Then, just like in bacteria, DNA ligase seals the nick.
So pol delta pushes, FEN1 cuts the flap, ligase seals. A different cleanup crew.
A different but equally effective cleanup crew for the lagging strand.
And now for the really unique eukaryotic problem. The ends, those linear chromosomes. You mentioned polymerase can't copy them completely.
Exactly. This is the famous end replication problem, or the telomere problem. Remember, DNA polymerase needs an RNA primer to start, and it synthesizes 5' to 3'. On the lagging strand, the very last RNA primer, right at the extreme 3' end of the template DNA, can be removed by FEN1. But there's no upstream Okazaki fragment to provide a 3' end for DNA polymerase delta to fill in that final gap.
So once the last primer is removed, there's a gap that can't be filled, leaving a bit of single-stranded DNA.
Exactly. And in the next round of replication, this shortened template will lead to a daughter chromosome that is slightly shorter. With each cell division, the chromosomes would get progressively shorter and shorter.
That sounds bad. Losing genetic information from the ends every time a cell divides.
It would be catastrophic eventually. Important genes near the ends would be lost. This progressive shortening is thought to contribute to cellular aging or senescence.
So how did nature solve this? There must be a solution.
There is a very elegant solution, the enzyme telomerase. This discovery earned Carol Greider and Elizabeth Blackburn, along with Jack Szostak, the Nobel Prize. Eukaryotic chromosome ends have specialized structures called telomeres. These consist of short, tandemly repeated DNA sequences. In humans, the repeat is TTTG, and a characteristic 3' single-stranded overhang.
TTT, repeated over and over.
Yes, hundreds or thousands of times. And telomerase is the enzyme responsible for maintaining the length of these telomeres. What's unique about telomerase is that it's a ribonuclear protein. It contains both protein subunits and an essential RNA component called TARF. Telomerase RNA component.
It has RNA built into it.
Yes. And crucially, this Turk RNA contains a sequence that is complementary to the telomeric DNA repeat sequence, like AAUCC in humans, complementary to TT.
Okay, so how does it use this RNA?
It uses it as a template. Here's how telomerase works, typically in 3 steps. 1. Binding. The telomerase enzyme binds specifically to the 3' overhang region of the telomere. The Turk RNA within the enzyme actually base pairs with the end of the DNA overhang.
Okay, it lines itself up using its RNA.
2. Polymerization. Then the protein part of telomerase, which is a special type of DNA polymerase called a reverse transcriptase, Turk telomerase, reverse transcriptase, uses the Turk RNA sequence as a template to synthesize new DNA. It extends the 3' end of the DNA strand, adding 1 repeat unit. For example, TTG.
Wow, it's using RNA to make DNA. That's reverse transcription.
Exactly. That's why it's a reverse transcriptase. It adds a short DNA sequence copied from its own internal RNA template. 3. Translocation. After synthesizing 1 repeat, the telomerase enzyme shifts or translocates down the newly extended DNA strand by about 6 nucleotides, realigns its RNA template, and repeats the polymerization process. Add another repeat.
So it binds, extends using its RNA template, shifts, binds, extends, shifts.
Yeah.
Over and over.
Precisely. This cycle can happen many times, significantly lengthening the 3' end of the chromosome. This added DNA acts as a buffer, ensuring that even with the end replication problem removing a bit each cycle, the crucial coding parts of the chromosome are protected.
It's essentially adding disposable, repetitive DNA sequences to counteract the shortening. That's incredibly clever.
It is. It solves the end replication problem for linear chromosomes.
And this ties into aging and cancer, you mentioned.
Yes, it has profound connections. In most of our normal somatic cells, non-reproductive cells, telomerase activity is actually quite low or absent after early development. As these cells divide throughout our lives, the telomeres do tend to shorten progressively. When they become critically short, it triggers a cellular response, often leading to senescence, the cell stops dividing, or apoptosis, programmed cell death. This is thought to be a protective mechanism against uncontrolled cell division and a contributor to the aging process.
So telomere shortening acts like a sort of cellular clock.
In a way, yes. A limited number of divisions before the clock runs out. Now, contrast this with cancer cells. A hallmark of many cancer cells is their ability to divide indefinitely. And in the vast majority of human cancers, maybe 85–90%, telomerase activity is found to be reactivated or significantly upregulated.
Ah, so they turn telomerase back on.
Yes, this allows them to maintain their telomere length despite rapid division, overcoming the senescence barrier and achieving cellular immortality, a key feature of cancer.
So targeting telomerase could be a way to fight cancer.
It's definitely a major area of cancer research. Developing drugs that inhibit telomerase is seen as a potential therapeutic strategy, hoping to selectively stop cancer cells from dividing indefinitely without harming normal cells too much, though some normal stem cells do rely on telomerase. It's promising, but like many cancer therapies, there are challenges and potential side effects to consider.
Fascinating. The basic mechanics of DNA replication leading to these huge implications for health, aging, and disease.
Absolutely. It connects the most fundamental molecular process to some of the biggest questions in biology and medicine.
Outro. Wow. Okay, that was quite a journey through the intricate world of DNA replication, from that elegantly simple concept of a template strand guiding a new one to the incredibly complex, almost Rube Goldberg-like choreography of dozens of specialized proteins working together. It's truly mind-boggling when you think about it happening constantly in our bodies.
It really is. And understanding these mechanisms, you know, the semiconservative nature that Meselson and Stahl showed us, the challenges of dealing with linear chromosomes, the ingenious solution of telomerase. It doesn't just illuminate life's fundamental processes, it also sheds critical light on areas like aging, genetic diseases, and cancer.
Yeah, and reflecting on how we even figured this stuff out, like the Meselson-Stahl experiment using heavy nitrogen, or the power of finding those temperature-sensitive mutants in bacteria, it really highlights the ingenuity involved in scientific discovery, just chipping away at the puzzle.
Definitely.
What really stands out to you from this deep dive, thinking back over it all, for me, I think it's just the sheer combination of speed and accuracy. How something so incredibly tiny and, you might think, fragile as a DNA strand gets copied billions of times with so few errors. It's the bedrock of life, isn't it?
It truly is. And it leaves you with some big questions, too. For instance, thinking about telomeres, considering their critical role in both normal aging and in allowing cancer cells to become immortal. How might future therapies effectively target telomerase in cancer without causing unacceptable damage to our healthy stem cells that also need it? It's a really profound challenge for medicine and research moving forward. How do we manipulate such a fundamental process safely?
That's a great thought to leave our listeners with, a real balancing act with potentially huge rewards, but also risks. Something to definitely keep an eye on in future research. Well, we hope this deep dive into DNA replication, drawing heavily from Brooker's comprehensive text, has given you a powerful shortcut to being genuinely well-informed on this vital topic. Thank you so much for joining us on the deep dive, and a big thank you, as always, for being part of the Last Minute Lecture family. We'll be back soon with another fascinating topic to unpack.